Good morning, students. This is your teacher Neelam Elawat, and we'll continue our chapter, that is excretory products and their elimination. And this is our lecture number 2, dear students, and I hope you're revising the chapter with me. Okay, so be ready with your books and notebooks. We'll start with the chapter, and today we'll be dealing with a very interesting topic, that is your urine formation. Urine formation, how the urine is formed in your body. So basically there are 3 steps which are involved in urine formation. First is glomerulus filtrate. The very first step is called the glomerulus filtrate, dear students. Now in this process what happens, there is a filtration of blood by the glomerulus. Now can you tell me where is glomerulus present? Very good, it is present inside the Bowman's capsule. So glomerulus will filter the blood, okay. So the first step is called glomerular filtration. The second step is called reabsorption by the renal tubules. And I hope you remember the renal tubules. Yes. So renal tubule means your PCT, DCT, Henle's loop and collecting duct. So they all constitute your renal tubules. So the second step involves the reabsorption. And third is secretion. That is, the tubular cells, they will secrete hydrogen ions, they will secrete potassium ions, they will secrete the ammonia into the filtrate, right? So this will result into the secretion into the waste. Now coming to the first step, which is called the glomerular filtration. This is the first step. Now first step in the urine formation is basically the filtration of the blood. So in this particular, in very first step, the blood will be filtered, and this filtration of the blood will be done by the glomerulus. So who will filter the blood, dear students? Glomerulus, right? Who will filter? Glomerulus will filter the blood, right? Okay. On an average, if we talk about, so 100 you can say 1100 to 1200 of ml of blood is filtered. This much of the blood is filtered. Okay, so on an average, our kidneys, they are filtering 1100 to 1200 of the blood per minute, and this 1100 to 1200 of blood is approximately 1/5 of the total blood that passes through the heart. तो ये आपका 1/5 blood है जो हार्ट के थ्रू पास होता है. So kidneys, they filter approximately 1100 to 1200 ml of blood per minute, and which is approximately 1/5 of the total blood pumped by the heart per minute, right? So your glomerular capillaries, they filter the blood. They filter the blood through 3 layers. The very first layer is endothelium of glomerular blood vessels. You must remember these 3 layers. The first is endothelium of glomerular blood vessels. The second is epithelium of Bowman's capsule, and third is the basement membrane. Okay? This third is the basement membrane. Now this epithelium of Bowman's capsule, okay, this second layer, that is the epithelium of Bowman's capsule, it contains special cells which are called podocytes. What are those cells called? Podocytes. The cells are called as podocytes. What do we call those cells? These are podocytes, and these cells, they are arranged in a very intricate manner so that there is minute spaces are left, and these minute spaces we have called slit pores. Okay, what do we call the pores? The slit pores. So filtration, the blood is filtered through these minute pores, these slit pores, very, very minutely. And it is filtered in a manner that all the constituents of the plasma except the proteins can pass onto the lumen of the Bowman's capsule. Okay, ये इतना बढ़िया तरीके से, इतना फाइन तरीके से ये फिल्ट्रेशन होता है कि प्लाज्मा के जितने भी कंपोनेंट्स हैं, प्रोटीन्स को छोड़कर, वो Bowman's कैप्सूल से पास हो जाते हैं. एंड दैट इज़ वाई दिस फिल्ट्रेशन इज कॉल्ड अल्ट्रा फिल्ट्रेशन. वाई डू वी कॉल इट एज अल्ट्रा फिल्ट्रेशन, राइट? The amount of filtrate formed, okay, this waste which is formed is called filtrate. So the amount of filtrate formed by kidneys per minute is called glomerular filtration rate. Kidneys ने कितने blood को filter किया है per minute, that is the glomerular filtration rate, and it is also called as GFR. What it is called as? GFR. It is called as GFR. So that is approximately 150 ml per minute. So GFR is your glomerular filtration rate, GFR. So what is GFR? 125 ml per minute, that is 180 liters per day. So your kidneys, they are filtering approximately 180 liters of blood per day. Okay, and this GFR is controlled by juxtaglomerular apparatus. Now it is a very sensitive region. This is called as JGA. JGA is juxtaglomerular apparatus. Okay, what is it? It is a juxtaglomerular apparatus. Now it is a very sensitive region which is formed by a combination of DST, that is distal convolute DCT plus your afferent arteriole. Okay, DCT plus your afferent arteriole. So these two, they together form the juxtaglomerular apparatus. Okay, what they form? Juxtaglomerular apparatus. So your GFR is regulated by JGA, right? Okay, very good. Now coming to the second portion. See, just now we have discussed that your kidneys, they will be filtering 180 liters of blood per day. But we are only excreting 1 to 1.5 liters of waste. Isn't this surprising? हमारी जो किडनीज़ हैं वो लगभग 180 लीटर्स ब्लड को फिल्टर कर रही हैं और उसमें से हम यूरिन सिर्फ 1 से 1.5 लीटर ही एक्सक्रीट करते हैं. So what about the rest of the material? So this means that 99% of the filtrate is reabsorbed back. Okay, 99% of the filtrate, it is reabsorbed by the renal tubules, and this process is called reabsorption. Okay, this process is called reabsorption. The tubular epithelial cells in different segments of the nephron. Okay, which cells? The tubular epithelial cells in different portions of the nephron. They either actively or passively, they absorb the substances like glucose, amino acids, sodium ions. So such materials, they are reabsorbed actively. Okay, they are reabsorbed, whereas the nitrogenous waste, they are absorbed by the passive transport. So most of the water is also reabsorbed, right? Is it clear? And during urine formation, the tubular cells, they also secrete substances like H plus ions, as I told you in the very beginning. The third step is called tubular secretion. So your tubular cells, they secrete the hydrogen ions, the potassium ions, and the ammonia. So tubular secretion is also a very important step in the urine formation because it helps in the maintenance of ionic and acid balance of the body fluids. Okay, it helps in maintaining the ionic and the acid balance. Okay, it helps in maintaining the ionic and acid balance of the body fluids. Now coming to the functions of tubules. Okay, what are the functions of the various tubules? Like your PCT. PCT means your proximal convoluted tubule, Henle's loop, the distal convoluted tubule, and the collecting duct. Let's look at their function. Now I hope you can see the diagram. Is this diagram clear to you all? So let's come to the PCT first. Now this PCT, it is lined by cuboidal epithelium. It is lined by cuboidal epithelium. And because it is lined by cuboidal epithelium, the surface area for reabsorption will increase. Okay, so essentially all nutrients, see, just look at this portion only. So all the nutrients, the bicarbonate ions, the sodium chloride, water and potassium ions, they are reabsorbed. Okay, they are reabsorbed in the PCT. Okay, while the hydrogen ions and the ammonia, they are secreted into the waste. देखिए आपका हाइड्रोजन और अमोनिया जो है वो ऐड किया जा रहा है, जबकि ये सारे पोर्शंस आपके रीएब्जॉर्ब किए जा रहे हैं. सो वेस्ट में से ये ब्लड में वापस रीएब्जॉर्ब हो जाते हैं. सो दिस मीन्स योर PCT, it helps in maintaining the pH and ionic balance in the body. Okay, then coming to your descending limb of the loop, coming to your Henle's loop. Now this portion is your Henle's loop. Okay dear, this portion is your Henle's loop, this thin portion, this is your Henle's loop, this thin portion is your Henle's loop. Now let's look at the Henle's loop. What happens in the Henle's loop? The descending limb of the loop is permeable to water. ये जो हिस्सा है, this is called descending limb of Henle's loop. It is permeable to water, so most of the water will also be reabsorbed. But it is impermeable to electrolytes. ठीक है, इलेक्ट्रोलाइट्स को impermeable है. क्लियर है? तो जैसे जैसे फ्लूइड नीचे जाता जाता है, वाटर एब्जॉर्ब होता जाता है और आपका यूरिन कॉन्सेंट्रेट होता चला जाता है. Now in the ascending limb, ascending means the one which is going up, this portion, this is the thin segment of the ascending limb. So the ascending limb is impermeable to water. ये वाला जो portion है ये impermeable है water के, right? And it will only allow electrolytes to move actively or passively. So आप देखिए आपका NaCl absorb हो रहा है, यूरिया वापस पोर किया जा रहा है, यूरिया is being added and sodium chloride, it is being absorbed. Okay, now coming to your distal portion. Now come to this portion, this portion. This portion is your distal portion. Okay, this portion is your distal portion. Now here there is conditional reabsorption takes place. Conditional reabsorption will take place. Like see, sodium chloride is reabsorbed, water again reabsorbed, bicarbonate ions they are also again reabsorbed, while potassium and hydrogen ions they are being added into the waste. ठीक है, तो आपके पोटेशियम आयंस और आपके हाइड्रोजन आयंस वो वेस्ट में ऐड किए जा रहे हैं. ठीक है, और सोडियम क्लोराइड, वाटर और बाइकार्बोनेट्स आयंस ये रीएब्जॉर्ब हो रहे हैं. ओके, नाउ फाइनली यूरिन विल कम टू योर कलेक्टिंग डक्ट. नाउ दिस कलेक्टिंग डक्ट extends from the cortex into the inner portion of the medulla. ठीक है, आपका ये जो कलेक्टिंग डक्ट है ये कोर्टेक्स में भी मौजूद, कॉर्टेक्स पोर्शन में और मेड्यूलरी पोर्शन में भी मौजूद होता है. नाउ हियर यू कैन सी लार्ज अमाउंट ऑफ वाटर इज एब्जॉर्ब्ड. यहाँ ज़्यादातर जो पानी है वो एब्जॉर्ब कर लिया जाता है. सो द यूरिन विल बिकम हाईली कॉन्सेंट्रेटेड. ओके, राइट, सो यूरिन विल बिकम हाईली कॉन्सेंट्रेटेड. So this segment allows the passage of small amount of urea into the medullary interstitium. यहाँ से थोड़ा बहुत यूरिया जो है वो मेडुला के इंटरस्टिशियम में चला जाता है. So thus it again plays a very important role in the pH and ionic balance of the blood. तो ये कलेक्टिंग डक्ट जो है आपका आयनिक बैलेंस मेंटेन करता है, pH को मेंटेन करता है. Clear everyone? So we can sum up in PCT. Most important your nutrients, 70 to 80% of electrolytes and water are reabsorbed. Henle's loop, osmolarity is maintained. Conditional reabsorption of sodium and water is there, and pH is maintained and the sodium potassium balance is maintained. Collecting duct में आपका large quantity of water is reabsorbed so that the urine can be concentrated. Now coming to the mechanism of concentration of urine. ठीक है, mammals have a ability to produce a highly concentrated urine. Concentrated urine means which contains least amount of water. जितना नेसेसरी पानी होना चाहिए उतना ही पानी यूरिन में एक्सक्रीट होगा. ठीक है, तो मैमल्स में एक यूनिक एबिलिटी है कि दैट दे विल प्रोड्यूस अ कॉन्सेंट्रेटेड यूरिन. द हेनलेज लूप एंड वासा रेक्टा दे प्ले अ वेरी इंपॉर्टेंट रोल. सो दो ऑर्गन्स हैं जो आपके कॉन्सेंट्रेशन में रोल प्ले करते हैं. द वेरी फर्स्ट इज योर हेनलेज लूप. ओके, so first is Henle's loop and second is your vasa recta. These two organs, they will play important role. Now if you have noticed, there is a opposite flow in the two portions of the Henle's loop. अभी हमने देखा था कि जो Henle's loop है जो डिसेंडिंग है उसमें मूवमेंट जो है लिक्विड की नीचे की तरफ होती है, जबकि जो हमारा असेंडिंग लिम्ब है उसमें मूवमेंट जो है लिक्विड की ऊपर की तरफ होती है. So in both the limbs of the Henle's loop, the movement of fluid is anti-opposite, or you can say it is moving in opposite direction, and thus it forms a counter current. Okay, दोनों में ये है, it forms a counter current. The flow of blood through the two limbs of the vasa recta is also in the you can say also in the counter current pattern. Okay, it is also in the counter current pattern. Let's see this. देखिए ये हमारा हेनलेज लूप है. ठीक है, दिस पोर्शन इज़ योर हेनलेज लूप. इस पोर्शन में फ्लूइड नीचे जा रहा है, इस फ्लूइड में पोर्शन में एक लिक्विड आ रहा है. सो दोनों फ्लो ऑफ लिक्विड जो है वो अपोजिट है. ये वासा रेक्टा है, इसमें भी इस पोर्शन में नीचे जाएगा, इस पोर्शन में नीचे जाएगा. तो वासा रेक्टा में भी और नेफ्रॉन में भी दोनों में ही काउंटर करंट पैटर्न है. ठीक है, ओके. एंड द हेनलेज लूप एंड वासा रेक्टा दे आर इन द क्लोज प्रॉक्सिमिटी विद ईच अदर. ये दोनों ही एक दूसरे के बहुत पास हैं. सो द प्रॉक्सिमिटी बिटवीन द हेनलेज लूप एंड द वासा रेक्टा एज वेल एज द काउंटर करंट इन बोथ ऑफ देम, इट हेल्प्स इन इंक्रीजिंग द ऑस्मोल रेगुलरिटी टुवर्ड्स द इनर मेड्यूलरी इंटरस्टिशियम. ठीक है, ये दोनों ही ऑस्मोल रेगुलरिटी को मेंटेन करने में हेल्प करते हैं. नाउ दिस ग्रेडियंट दैट इज अप्रॉक्सिमेटली 300 osmoregularity per liter in the cortex and about 1200 milli osmoregularity per liter in the inner medulla is there. देखिए आप ध्यान से देखिए यहाँ पर medulla, this is cortex region. तो cortex region में जो osmoregularity है वो 300 है, राइट. और medullary region में जो osmoregularity है वो 1200 ml है. और ये osmoregularity किस वजह से मेंटेन है? क्योंकि दोनों ही लूप्स में जो है मूवमेंट फ्लूइड की अपोजिट है. ठीक है, दोनों वासा रेक्टा में भी और नेफ्रॉन में द मूवमेंट ऑफ द फ्लूइड इज इन अपोजिट डायरेक्शन. ठीक है, क्लियर है? ओके. नाउ दिस ग्रेडियंट विच इज मेनली कॉज्ड बाय दिस डिफरेंस, दैट इज हियर इट इज 300 ml, हियर इट इज 1200 ml. दिस इज मेनली बिकॉज ऑफ NaCl एंड यूरिया. ये जो डिफरेंस है ये सिर्फ NaCl और यूरिया की वजह से होता है. ठीक है ना. NaCl इट इज ट्रांसपोर्टेड बाय द असेंडिंग लिम्ब. अब आपका असेंडिंग लिम्ब देखिए, ये असेंडिंग लिम्ब है. ठीक है, असेंडिंग लिम्ब में देखिए NaCl ट्रांसपोर्ट हो रहा है. ठीक है, सो द असेंडिंग लिम्ब ऑफ द हेनलेज लूप विल ट्रांसपोर्ट yes, it will transport the NaCl. ठीक है, यहाँ से NaCl ट्रांसपोर्ट हो रहा है एंड दिस विल गो इनटू दी असेंडिंग पोर्शन ऑफ वासा रेक्टा. ठीक है, ये हमारा असेंडिंग पोर्शन है. असेंडिंग पोर्शन नेफ्रॉन के असेंडिंग पोर्शन में से NaCl वासा रेक्टा के असेंडिंग पोर्शन में चला जाता है. ठीक है, this means the NaCl is it is being returned into the interstitium by vasa recta. Similarly, small amount of urea enters the thin segment of the ascending limb. अब क्योंकि NaCl remove हो रहा है, so some amount of urea will enter into the ascending limb. So इसके exchange में आपका यूरिया एंटर हो जाता है और NaCl यहाँ से निकलकर वासा रेक्टा के थ्रू इंटरस्टिशियम में पहुंच जाता है. ओके, क्लियर है? नाउ दिस मैकेनिज्म इज कॉल्ड काउंटर करंट मैकेनिज्म. इस मैकेनिज्म को हम क्या कहते हैं? काउंटर करंट मैकेनिज्म. ओके, एक बार दोबारा देख लीजिए. कॉर्टेक्स में आपकी ऑस्मो रेगुलरिटी 300 ml है, 300 है। मिड्यूला में 1200 है, 1200। दूसरी बात, आपका जो असेंडिंग लिप है नेफ्रॉन का जो असेंडिंग लिप है, यहाँ से NaCl जो है वो जाता है वासा रेक्टा के असेंडिंग लिम्ब में एब्जॉर्ब हो जाता है। इन एक्सचेंज द यूरिया विल बी ट्रांसपोर्टेड इनटू द असेंडिंग लिम्ब ऑफ द हेनलेज लूप। क्लियर? एंड दिस mechanism is called as counter current mechanism. What do we call it as? Counter current mechanism. ठीक है? Okay. Now presence of this gradient, it basically helps, or it provides an easy passage of water from the collecting tubules, thereby concentrating the urine. अब आप देखिए collecting duct को देखिए, this is your collecting duct, this is your collecting duct, ठीक है। सो collecting duct में से देखिए majority of the water जो है वो remove हो रहा है, it is being reabsorbed, वापस से reabsorb किया जा रहा है, and because of this the urine will become highly concentrated. So the human kidneys can produce urine nearly 4 times concentrated than the initial filtrate found. ठीक है, जितना हमने फिल्ट्रेशन पहले किया था, अल्ट्रा फिल्ट्रेशन, उसका 4 गुना कम यहाँ पर फ्लूइड हो जाता है, यानी 4 गुना कॉन्सेंट्रेट हो जाता है, 4 गुना जो लिक्विड है, you can say 4 times liquid is absorbed and only a small fraction of liquid is left. So हम यूरिन की कॉन्सेंट्रेशन 4 गुना बढ़ा देते हैं, ठीक है। Now coming to your topic, dear students, first of all make notes of this topic. So you will beautifully draw the diagrams and you will make notes of this topic, and then you will go through the page numbers 293 and 295. Right everyone? So you will read the page numbers 293 and 295, and if there is any doubt you will certainly send your queries to me on my personal number. Okay everyone? So that's all for today. Bye everyone.